Hello, everybody. I'm Abby from Precisionary Instruments, and welcome to today's talk as part of our 2026 scientific series. This is the first one of the new year, and I'm really delighted to introduce today's speaker, Professor Youjin Lee, who is an assistant professor in the Department of Immunology at the University of Pittsburgh. So Dr. Lee's laboratory focuses on understanding how spatial organization within tissues shapes immune cell behavior and function. Her work combines precision-cut tissue preparation with single-cell spatial multi-omics and computational modeling, and really excitingly, machine learning approaches to uncover how complex tissue microenvironments govern immune responses. She's going to share with us some of her preliminary data on kidney and spleen today, and she will also share how spatially resolved single-cell data can be generated from intact tissue samples, and how these data can be modeled to reveal tissue-level interactions, and how computational frameworks can extract predictive insights from complex biological systems. So we're very excited to host you today, and please join me in welcoming Professor Youjin Lee. So with that, I'm going to hand it over to you, and I'm very excited to listen to your talk.
Thank you so much, Abby. So obviously, I want to thank the organizers for inviting me to give a talk at this webinar. As Abby mentioned, my name is Youjin Lee, and I'm an assistant professor in the Department of Immunology at the University of Pittsburgh. So with that, I will start off with my first slide. I can get this working. Okay, so to me, this is kind of an example of why I got interested in this in the first place. As you can appreciate, these are sections from 3 different tissues: brain, intestine, and tumor. They were not cut by a compressome, but I think they illustrate a very valid point, which is that even though they're very distinct tissues, they all have a distinct, intricately organized structures. Even tumor, which, as you know, kind of arises from a single transformed cell, does not really grow chaotically, but also in an organized manner, as you can see from the right slide here. So what do we do with this? So our lab is, on a nutshell, a tool building lab. We work on spatial transdomics, so experimental tools that we develop to try to enhance how we can survey the environment better. And then we can also build machine learning tools, so a different type of tool, computational tool that allows us to interpret the data that we generate from the spatial transdomics data. And all this is to try to understand the cell-cell communication networks within the tissue microenvironment that helps us decode what are some of the elements that drive health and disease. So to give a little bit more granularity to what I'm going to be presenting to you today, so in particular, I'm actually going to be talking to you about how we actually build a specific tool by combining spatial transdomics tool with a CRISPR perturbation screen. And then we also, this is kind of, as I mentioned, kind of the experimental component that I'll be talking to you about today. But at the same time, we're also going to be kind of doing a little bit of a dive into how we can generate a machine learning perturbation model as well by utilizing the data that we generate from spatial transdomics. So here is kind of the outline. The first part is how we use a compressome to generate the single-cell spatial transdomics data. And then the second part is then taking the data set that we generated from our platform that I'm going to be mentioning to you later on, as well as commercially available platforms. And we apply this data to generate a machine learning model that we're calling Space Traveler. And the name will become very obvious to you as I do a little bit of a dive into that. Okay, so here's my disclosures. Okay. So while I was a postdoc at UCSF, I developed this new novel platform that we called XYZ. XYZ literally stands for XYZ spatial coordinates combined with spatial sequencing. And then this will allow us to better understand how individual cells function within the anatomical space by linking spatial metadata with single-cell RNA sequencing. So that was version 1, but since I've started my own lab, I have developed a version 2, which then allows for us to integrate multi-omics with our single-cell spatial sequencing platform. And then the key element here is that the fact that this platform generates true bona fide single-cell data, and I'm going to show you evidence of that later. So just to kind of give you a brief overview for how this platform actually works, what we do is we have this robot that deposits the spatial oligos into these very tiny microwells. We cut the tissue, fresh tissue. So this is where the compressome comes in, where we're able to slice various tissues. And we've actually tried kidney, liver, brain, lung, spleen, as well as lymph nodes and tumors. So, but the data I'm going to be showing you today is mainly derived from the kidney and the spleen. But what we do is we cut the fresh tissue, and then we place the tissue on top of this array where it has been spotted with these spatial oligos. The spatial oligos get transferred over onto the tissue. And then once that has happened, we take the tissue and then we digest it into true single cell suspension, and we run it into a commercially available single cell RNA sequencing platform. In this case, what we like to use in the lab is 10x. And then because of the fact that 10x, as most of you may already know, can already incorporate single cell multi-omics, because of the fact that we've just did a barcoding of the spatial oligos earlier prior to us piping it into the 10x platform, we're now able to integrate both the spatial component with single cell RNA sequencing. So this is kind of how XYZ version 2 works. So when we generate the data, and this is just an example of the type of data we're able to generate, this is from the spleen data. You're able to get spatial data that is bona fide single cell data. As you can see here, this is a UMAP of the various cells that were found in the tissue. But not only are you able to generate specific subtypes, so dendritic cells and versus T cells, but because it's really good quality single cell data, you're now able to dig deep a little bit into the dendritic cells and be able to isolate the various subpopulations that are present, in addition to, for example, different cell states that are associated with T cells. So early effector cells or helper T cells can also be gleaned from this data. And because of the fact that everything is spatially tagged, and we can also pair with multi-omics. Here's an example of how we actually generate multi-omics data. This is one example of how we now pair protein. So this is an example of a UMAP where now instead of cells and RNA, what we've actually overlaid is the protein expression of, in this case, CD45. And then, as I mentioned, since every cell is spatially tagged arranged in the wells, you can now see an arrangement of the varying degrees of CD45 protein expression across the spleen tissue as well. So this is an example of how we're able to generate single cell spatial data using the tissue that we generate off of the compressome. Okay, so to kind of get into the meat of this, as I mentioned, I'm going to be talking to you. The first part is how we use the compressome data to generate this unique spatial data that we have in the lab. And here is one type. This is sort of what it looks like once we generate when we take a slice of the kidney, and using speed 5 and oscillation 3. I had to get this information more recently, but yeah. So this is sort of the kind of the kinetics and the speed in which we actually generate or cut the tissue, and then when we actually put it on the array, here's an image of the kidney on the array. And because of the fact that we were able to generate single cell data, as you can see, we can actually map the cells, each individual cell, back into the space. And this is just sort of how we merge the two information together, our single cell data in addition to the well data with the tissue that we have on the array. And just to give you an idea for the quality of data we're able to generate using this platform, is you can see that the quality of data is very, very high. This is now compared to the annotated data that was generated by the Cell Atlas, and every single cell type was able to get discovered from our spatial sequencing platform. And this is compared to the one that has been annotated by a foundation, so CZI. So, and I want to just, I guess, just mention that the reason we're able to generate this high quality data is because we actually start off with fresh tissue, which compressome allows us to do. And as a result, it's essentially single cell data that have now been spatially tagged. So as I mentioned, since they are spatially tagged, I can show you kind of a snippet of sort of what that will look like. Here is a UMAP of all the different cells that we derived from the kidney slice that I showed you earlier. And one thing I want you to pay attention to is actually the myeloid population, which ends up being a huge population, as you can see from the healthy kidney. And then when we're able to kind of determine the different cell states, we're able to identify two distinct myeloid populations that we're just naming as MAC0 and MAC2. No, no reference at all to what they're actually named. We just went through 0, 1, 2, 3 are the 4 types that we found, and I'm just showcasing you two of them. But as I mentioned, since every single cell can be localized to a region in the tissue, when we actually plotted these MAC0 and MAC2, here's what we found, that somehow or another, it seems like this MAC2 population seems to occupy the outer space of the tissue, and the MAC0 seems to occupy the inner core of the kidney. So because of the fact that we're able to generate this very interesting single cell true spatial data, we can see that they are not only are we able to get the various information from the cells, from the single cell itself, but at the same time, we can actually see the distribution, a unique distribution as it lays within the kidney architecture as well. So this is one example of that. Another thing I wanted to mention is, as I mentioned, kind of going into kind of the multi-omics component. This is a thing that is very preliminary that we're generating in with Survey Genomics in collaboration. So here's sort of kind of the layout. Our institute wanted to actually understand the gene knockouts of hematopoietic stem cells, differentiation and spatial distribution of the secondary sites of hematopoiesis. And so you have a Cas9 CRISPR cells that have a guides that insert into them, you transfer them in, and then you look at the various organs. And what you can see here is that here are the examples of the kidney, sample 1 and sample 2. You're able to see not only the positive stain of the spatially tagged samples, but at the same time can also see the cells that were spatially tagged are also CRISPR edited. And then we're able to annotate all the different cell types that were found. In fact, across the 4 samples, we were able to recover 62,000 cells and 58,000 cells after filtering of various different types of unique cell states and types. And this is just a kind of QC metrics to kind of illustrate to you that the 10 genes that we put in, 73% of the cells were positive for at least one gene guide set, meaning that we're able to now identify and detect them from our spatial sequencing platform XYZ. But here's what I think is the coolest part of the data that I think has never been shown before. The fact that you can see that each of these guides that have been edited, here's one guide, here's another guide here, or the gene associated with the guides, they seem to occupy different regions within the space. So what we're actually capturing here is that the guide distribution, the cells that have been edited by this particular gene are co-localizing into distinct regions within the kidney. And as you can see, depending on the gene that's been edited, it seems to modify the behavior of the cells that have been transferred in and then occupying different regions or niches within the kidney architecture. So this is kind of a really interesting thing that we're able to discover, and we're actually pursuing further analysis from this type of data. Okay, so that was kind of how we actually use the data to generate single cell spatial data. I want to now kind of dovetail into the second part, is how we actually apply the data. As I mentioned, this thing that we have generated in the lab, we're calling a space traveler. It's an in silico prediction of genetic perturbations across the tissue microenvironment. It is in collaboration with Jishnu Das lab and Amanda Pollock's lab and spearheaded by 2 very talented graduate students. So just to kind of give you an idea for why we thought this is important. So as I mentioned kind of in the first slide, we're really interested in wanting to understand what happens within the tissue microenvironment. And this is kind of a schematic of that, where each cell, as you can imagine, is embedded within the microenvironment, and they all express various genes that reflects not only their identity, but their functional states. However, you can also probably imagine that the genes and the cells that are expressing these genes are not working in silos. So they are not operating in isolation, but in fact, they're likely shaped by the interactions of the neighboring cells and influenced definitely by the environmental cues that the cells are embedded in. And on the right, as you can see, the consequence of this behavior, it would be various different cell states that we refer to as kind of lineage trajectories that people do further analysis on. So our question when we first started to kind of think about how we can apply the spatial data is, we wanted to ask, how do these local environmental cues and gene networks steer a cell towards one cell state over another? And we came up with this space traveler, and the reason we're calling it space traveler is because it's spatial for the S, but then we incorporate 3 different components from the data that we generate, is the transcription factors, ligands, and the receptors. And then by layering these informations together, we can ask, how does a signal from one cell influence a gene expression in another? And how do these shape cell states and fate, as I mentioned before? And the reason we are interested in kind of using these three parameters, the ligand, receptor, and transcription factors, is because of the fact that if you kind of think about it, the transcription factor kind of tells you what are the genes that are being regulated or turned on that modifies the behavior of that cell. And that is kind of the intrinsic component of the cell. But then when you incorporate the ligand and receptor information from that cell, and that information is embedded within the spatial architecture, what you can start to understand is kind of the cell-cell interaction and how the cells are influencing each other within that environment. So with the information combined, we're able to now generate kind of the spatial distancing map, and then be able to sort of understand how the cells are working in relation to each other of the various parameters that we actually put in as inputs and outputs. So when we actually have this machine learning model learn the cell-cell relationship within that tissue, here's something that we're able to actually discern. So this, to me, by far is kind of the coolest data that my student actually showed me. So here's again the image of the kidney slice that with the various cells that were able to generate using the compressome slice, in addition to the spatial transcriptomics transcriptome data that we're able to generate. And then we apply the machine learning model that we've able to use to train the data set from. And what I thought was super cool was the fact that our model does not know the data is actually originated from a kidney. But what it had learned is that it's learned the different cell-cell interactions. And to me, the most important thing is it's learned the edges. So these are kind of neighborhoods where it noticed that within each well, the neighborhood or the edges of one type of cell changes to another, or the transcriptome changes. And as a result, is able to draw out where the boundaries are of the various tissue neighborhoods. But what I thought was really cool was that even though we just had it train on the transcriptome data from this tissue slice, it was able to generate what ended up looking like a kidney. So without knowing that it didn't say that it was a kidney, but somehow, because it's learned the cell-cell relationship within the tissue and the transcriptome data, the output ended up becoming a kidney, which I thought was a really cool component of this. And so a lot of information can actually be gleaned from the transcriptome data if there's great quality to begin with, but at the same time, if you can apply some of these really interesting machine learning models. But I wanted to kind of give you an idea or at least to give you a snippet of kind of a validation that we realize that our machine learning model may actually be effective in being able to predict. So this is not trained on compressome data, but actually publicly available data. It's a human tonsil. And as you can see, if you generate the different populations out of this spatial data, you're able to see the different T cells, B cells, the neritic cells, germinal center B cells, and the plasma cells. And you can generate a UMAP of that. And because of the fact that this single cell data was generated from a spatial data, you can then map each of these cell populations to a 2D spatial space, as you can see here. The black here is outlined by the germinal centers that are present due to the germinal center B cells. So what we did was then we started to compare within the germinal center the various genes that are highly expressed between one versus the other. So in germinal centers, there is a key element where you have a dark zone and a light zone that are associated with the germinal centers, and that basically just is determined by the function. And when we actually compare these two populations of the single cell data, and then we try to see what are some of the key differentially expressed genes between these two populations, we've identified one called FOXO1. And here you can see that when we now this is computational deletion. Once we've actually had our machine learning model learn and train on the spatial single cell data. And what we found out is that when you computationally delete FOXO1 in the germinal center B cells, the model started to predict that the germinal center B cells will become more like from a dark zone to a light zone. So this is it's not arrows showing movement, but in fact, it's just kind of trying to illustrate that once you delete FOXO1 computationally in the germinal center B cells, what you see here is that they become the germinal center B cells that are residing or seem to be more like a dark zone germinal center B cell become more like a light zone in the germinal center. So this is what it's predicting, and as well as predicting that there becomes this intermediate zone as well. But when you map that spatially, you can see that it essentially, you have a before FOXO1 knockout and an after FOXO1 knockout. The red here is the dark zone, and then the green here, as I mentioned, is kind of the light zone. And what is the model is predicting here is that before FOXO1, you see the red and the green divided into two dark zone and light zone within the germinal center of the tonsil. But then the model predicted that after FOXO1 knockout, all of a sudden the dark zone disappears and becomes more like intermediate to light zone. This is what the prediction is. Now, what is really cool is then we went back into literature and we found that in 2017, somebody's actually already done this experiment. So they identified that a transcription factor FOXO1 controls germinal center B cell proliferation and response. And as you can see here in this study, they demonstrate that the ablation of FOXO1 after germinal center development led to the loss of dark zone germinal center B cells and the disruption of the germinal center architecture. So what is really cool is that once you actually have spatial data, really quality spatial data that is, in this case, a single cell, single nuclei level, and you're able to now train the model to have it learn the cell-cell relationship within that tissue architecture. You can essentially perturb a gene in space, any gene of interest, whether as long as it's a transcription factor, ligand, or receptor. And you can then have the model predict the outcome of that. So, and we can see that it's pretty accurate because, at least in this particular case, because our model predicted exactly what this particular study in JXMed has done back almost like 8 years ago. So, and in our study, we show many different examples of this, but this is, I think, one very nice classical one that showcases the power of machine learning, and then at the same time, the power of using good single cell spatial data to be able to train some of our machine learning models to generate different predictions. Okay, so that is actually what I have to present to you guys. Just to kind of recap, I introduced to you guys XYZ version 2. It is a novel single cell spatial, what I would like to call multi-omics platform that integrates proteomics, and I also showed you a little bit of that. It's the integration of also CRISPR perturbation as well. That is experimental version of it. But at the same time, by using and training on just regular single cell spatial data, what you can also do is generate a machine learning model, and so an in silico perturbation model, and kind of a CRISPR perturbation model that is spatially aware. So I also introduced to you for the second part of how we actually utilize the spatial data to generate a machine learning model that we like to call Space Traveler. So that is all I have for you today, and I want to acknowledge the people who did the work. So this is Kushal, who's the lead author in the paper, and then we have Ali Wong from Jishnu's lab, and these two teamed up to generate Space Traveler. And then the rest of the lab does a lot of the wet lab of generating the spatial data, and they probably know how to use the compression better than me. But yeah, they're the ones who are actually generating some of the spatial data that we're using now for additional machine learning models. And I'm happy to take any questions. That's it. Thanks.
Wonderful. Thank you so much. What I want to say as we conclude this talk is first and foremost to thank Professor Lee for your work and for sharing your information with us. It's very exciting to see the preliminary data in the different organ systems. And it's amazing that you tried it in all sorts of organs already.
Yes,
which is
we
really
have
neat.
actually. Yes.
Yeah. And I also wanted to conclude by saying that for our attendees, if any of you want to have questions later on or want to connect with Professor Lee, please let us know, and I'm happy to facilitate the connection. And if you have questions about using the compressed for making precision cut tissue slices for your own research, let me know directly and I'll be able to assist. And with that, one final round of applause. Thank you very much. And please stay in touch. Thank you, everybody, for attending.
Thank you so much. Bye. How do I do this? Just leave it.